The main difference in the specifications is that in the phase 1 IND, we are giving tentative acceptance criteria, while in the case of phase 2 and 3, the specifications are getting firmed up and we are expected to generate specifications with list of tests, analytical procedures and acceptance criteria. And these need to be based on multiple pilot or full-scale batches and with appropriate statistical analysis. Then coming to the analytical procedure, scientifically sound analytical method, and here full validation is not required. While for phase 2 and 3 IND, full validation is required for the new drug application, which means when we are submitting the new drug application, we must have full validation of the analytical method, and this should become part of the CMC section in that particular IND. Coming over to the stability data, here preliminary data is to be submitted, while in phase 2, stability indicating method-based data need to be provided. And in phase 3, a detailed stability protocol and stability data should be provided. So you can see that there is a gradual progression in the extent of the data, which is included in the CMC section. Here only preliminary data is requested, while here stability indicating method, and in phase 3 there is detailed stability protocol based data that is required. For container closure system, we require the information to be given in phase 1 IND as well as in the phase 2 and 3 INDs. Then we move over to the information related to the drug product, which is the formulation. First part is the components and quantitative composition. So list of all components with reasonable alternatives for inactives can be included. So the interesting part here is that there are reasonable alternatives which are allowed for the excipients or the inactives. And we need to include all components that will appear and those that will not appear but may be used in manufacturing. By this, what we mean is, say for example, we are using water for granulation, that is wet granulation, but in the drying step, the water will be removed from the product. So this means that the product will not be present in the final product, but it has been used during the manufacturing. So purified water needs to be mentioned in the list of components, and you can mention in front of this that used for the purpose of wet granulation, not present in the final product. Then a quantitative composition with reasonable variations and milligram or ml per unit dosage form composition and batch formula needs to be provided. Quality of the components, and for novel excipients, additional manufacturing information might be required. If you are using one excipient for the first time, so it will become a novel excipient, and the regulatory agencies want you to supply additional information about that particular excipient. In fact, it is interesting that for a new excipient, safety information to the same extent as is generated for a new chemical entity needs to be provided. So this is a different part, that for the novel components or novel excipients, additional manufacturing information may be required. Similarly, in phase 2 and 3, provide details of changes over phase 1. Unit formula and batch formula needs to be provided, and formulations of certain drug product delivered by devices. For example, metered dose inhalers, dry powder inhalers, and nasal sprays should be similar to that intended for the marketed drug product. Now, why there are special requirements for these dosage forms? is because of the fact that the device that is used in MDIs, DPIs and nasal sprays is critical for the functioning of the dosage form. So in the case of DPI, the delivery of the product will be affected by the device that is being used. So the regulatory agencies want that in the clinical trials, same device which is going to be ultimately marketed should be used for conducting the clinical trials. Otherwise, if you change the device in between the clinical trials, then the entire data is likely to change and the clinical outcome of the study is expected to change. So to avoid those complications, it is required that the same device that is going to be used in the marketed product should also be used for conducting the clinical trials. Specifications of the components, that is excipients, is required in the phase 1, and in phase 2 and 3, changes in acceptance criteria and grade need to be highlighted. Information for excipients not included in previously approved drug products should be equivalent to that submitted for the new drug substance. I just mentioned this part, that if you are using a new excipient which has not been approved in any previous drug product, then the information of safety which is to be given for the new excipient is of the same level, that is equivalent to that of the submitted or that is submitted for a new drug substance. So you need to provide all the safety related information that the new excipient which you are using is going to be safe for use in the human subjects. So we need to provide very detailed information for the excipient as well. This is the reason why there are so few excipients that are introduced in the pharmaceutical markets, because the amount of money and time that needs to be spent on a new excipient is very high, because you have to conduct similar type of safety studies as you will conduct for a new chemical entity. In the drug product also, the name and address of the drug product manufacturer needs to be given. And in the phase 2 and 3, any update on phase 1 details should be given. For specification, brief description of the proposed acceptable methods to ensure identity, strength, quality, and purity of the product should be provided. Certificate of analysis of the clinical batch and validation and established specifications are not required at this stage. So this is the difference between phase 1 and phase 2 and 3 information, that in phase 1 validation and established specifications are not required. In phase 2 and 3, you have to report changes over phase 1 specifications. Chemical tests like disintegration, dissolution, assay, content uniformity, and microbiological test like sterility, LAL test for the pyrogens added or deleted over phase 1 should be reported. Analytical method with validation report should be available if requested, and degradation profile should be provided. So here the requirements for the specifications become more elaborate in the CMC section of phase 2 and 3 INDs. Next one is the manufacturing and packaging related information. So a flow diagram giving brief description of the manufacturing and packaging should be provided, while in the phase 2 and 3 also a similar way of representing the information should be used. Then we come to the container closure system or the packaging system. So in phase 1, brief description of the CCS should be provided, while for phase 2 and 3, for packaging components with compendial standards, for example, glass, polyethylene containers, compliance with appropriate compendial standards should be stated. If the sponsor refers to information in a type 3 DMF, an authorization letter from the DMF folder should be provided. container closure systems for the meter dose inhalers, dry powder inhalers, and nasal sprays should be similar to the marketed product. This we had highlighted earlier also, that the container closure system of MDIs, DPIs, and nasal sprays in the clinical trials should be similar to that of the marketed products. Moving over to the stability related information. In the phase 1 IND, in the CMC section, detailed data and protocol is not required. Data on representative material may be submitted, and data to support the duration of the clinical trial is required. So if the clinical trial is only for 1 month period, you require stability data which is suitable to justify 1 month of shelf life. Similarly, if your clinical trial goes for 3 months, stability data should be sufficient to support 3 months of the clinical trials. Then the protocol in the phase 2 and 3, protocol including list of tests, analytical procedures, sampling time points to be provided. Stability data of phase 1 and photo stability data should be provided, and full data to support the shelf life of the product should be provided. So this is an important component, that the data to support the shelf life should be provided. Here the requirement was only to support the duration of the clinical trials, while here the requirement is to support the entire shelf life. Process validation is not required for the drug product in phase 1, while it can be completed after new drug application approval for the phase 2 and 3, which means the product validation or the process validation can be carried out after the new drug application approval. For placebo, the information should be similar as possible to the active dosage form in physical characteristics and identically packaged. Brief information on composition, manufacture, and control of the placebo is required. And here data showing absence of active material should be provided. For labeling, a mock-up label needs to be provided in the phase 1, while for phase 2 and 3, update over phase 1 should be provided. There is another information related to the environmental assessment, which is the impact of the manufacturing of the product on the environment. In US FDA, it is essential to provide the statement of environmental assessment. So the company can provide a categorical exclusion in case of the most drugs by providing relevant information. And what we mean by categorical exclusion is a categorical exclusion is a category of actions that the agency has determined based on past experience do not individually or cumulative have a significant effect on the human environment. As a result, neither an environmental assessment nor an environmental effect statement is required for these actions. The usual concerns in the CMC section in the phase 1 are that the emphasis here is on the information that allows evaluation of the safety of the subjects in the proposed study. So the main concern areas are product made with unknown and impure components, possession of a chemical structure of known or highly likely toxicity. The product cannot remain stable throughout the testing program. And then impurity profile with potential health hazard or insufficient impurity profile. The sponsor should be able to relate the drug product being proposed for use in clinical study to the drug product used in the animal toxicity studies. So this is a very important aspect, that the drug product which is going into the human clinical trials should be equivalent to the product which was used for the animal toxicity studies. Otherwise, the data that you got from the animal toxicity and that you are getting in the clinical trials will not be comparable. Hence, it is necessary that these two formulations should be equivalent. In phase 2 and 3, additional information as generated in phase 1 of clinical trials should be submitted. If the clinical trials begin with phase 2 itself, as is the case in some anti-cancer drugs, then all the concerns associated with phase 1 should be additionally addressed in phase 2 and 3. So this is the comparison of the CMC information as it is present in the IND phase 1, while comparison has been made to the information which is given in phase 2 and 3 IND. So this is very important from the perspective of a formulation development scientist, because they are the ones who contribute to the CMC information, and this table gives them an idea that what are the requirements of the CMC information in phase 1 of the clinical trials and phase 2 and 3 of the clinical trials. So with this, we finish our discussion on the information of the CMC information, and we will be moving to the next topic in our next class.